Welcome back, everyone, to another edition of the Explainer, specially created for St. Paul American School Clerk. Today, we're diving straight into the microscopic world with our exact production title, G9 Biology, Unit 5, Lesson 1, Bacteria and Viruses and Bacteria. Think of today's Explainer as an exploration of our microscopic roster. We're going to uncover the structural secrets of these tiny entities and figure out exactly how they operate, survive, and impact our world. Okay, here's our agenda for today. 1. Objectives. 2. The biology of bacteria. 3. Viruses and prions. 4. Gram stain analysis model. 5. Misconception and formative checks. And finally, 6. Concise lesson recap. Let's get right into it.
Section 1. Objectives. Setting the stage.
So here's exactly what you should focus on today. By the end of this, you'll be able to clearly distinguish between prokaryotic bacteria and non-living viruses. We're going to break down bacterial cell shapes, their arrangements, and their pretty incredible survival structures. We'll also analyze the gram stain process to see what it actually reveals about cell walls. And lastly, we'll define prions based on their unique structural folding propensity. Keep these goals in the back of your mind as we go.
Section 2. The biology of bacteria. Meet the prokaryotes.
Right, so the absolute crucial point here is understanding what makes a cell prokaryotic. These are simple, mostly single-celled organisms, and their biggest defining feature, they totally lack a membrane-bound nucleus. So instead of their DNA being neatly packaged away in a nucleus, their chromosomal DNA just kind of hangs out in a central region we call the nucleoid. Also, keep in mind, they're typically much, much smaller than the eukaryotic cells that make up plants and animals. Now, within the bacteria group, we've got two completely distinct types. First, there's archaeobacteria. The prefix archae literally means ancient. These guys absolutely thrive in extreme, harsh environments. We're talking boiling undersea vents or the super salty Dead Sea. And structurally, their cell walls actually lack a substance called peptidoglycan. Then, on the other hand, we have eubacteria, which are the true bacteria. These are the ones we interact with in our common, everyday environments. Unlike the ancient guys, their cell walls do contain peptidoglycan, and they play a massive variety of roles in our world, as you'll soon see. And naming these eubacteria, it's honestly like a microscopic puzzle where you just combine shape and arrangement. If a bacterium is round, it's a coccus. Rod-shaped, that's a bacillus. And curved rods are vibrios. Then you just look at how they arrange themselves. If they're in pairs, you slap on the prefix diplo. If you form long chains, kind of like a microscopic string of pearls, that's strepto. And if they group up in large bunches, it's staphylo. So if you ever hear the term streptococci, you instantly know you're dealing with round bacteria arranged in long chains. Pretty cool, right? But what happens when the environment becomes completely deadly for these bacteria? Well, some of them, specifically certain genera like Bacillus and Clostridium, have a really incredible trick up their sleeve called the endospore. An endospore is this dormant, insanely tough, non-reproductive survival structure. Think of it as a highly resistant biological bunker. When conditions get rough, like starvation, extreme heat, or chemical exposure, the active vegetative cell literally builds this endospore around a copy of its DNA. These spores can survive completely dehydrated for hundreds, maybe even thousands of years, just waiting for conditions to improve. Okay, so when conditions are actually good, bacteria need to multiply, and they do this incredibly fast through an asexual reproduction process called binary fission. It's a simple step-by-step. First, the bacterium copies its chromosome. Next, both the original and the copy attach themselves directly to the cell's plasma membrane. As the cell grows larger, it naturally pulls those two chromosomes apart. And finally, a partition forms right down the middle, dividing the organism into two completely identical smaller cells. Boom. Multiplication complete. You know, it's so easy to just think of bacteria as germs, but we really have to ground this biology in reality. The vast majority of eubacteria are incredibly helpful. I mean, we completely rely on them for nitrogen fixation to fertilize our agricultural fields. They're essential for making foods like cheese and yogurt, and they even help us create life-saving antibiotics like streptomycin. But yes, of course, there are the harmful ones too, the pathogens. And those are responsible for diseases ranging from strep throat and tetanus all the way to Lyme disease and cholera. Section 3. Viruses and prions, the non-living roster. Now, here is a definitive, absolutely crucial statement for you. Viruses are non-living. Period. They don't grow, they don't develop, and they do not undergo cellular respiration. A virus is essentially just a biological package. Structurally, it's just an outer protein coat called a capsid, and inside that coat is a small amount of nucleic acid, which can be DNA or RNA. Because they completely lack basic cellular machinery, they are what we call obligate parasites. They absolutely must have a host to do anything at all. So, how exactly do they exploit that host? They use a process known as the lytic cycle. Step 1. The virus attacks and binds to a specific receptor on the host cell. Step 2 is the infiltration phase. The virus injects its nucleic acid, its genetic code, directly into the host. And finally, in step 3, that injected code literally hijacks the host cell's machinery, forcing it to just multiply the virus parts. Once they're assembled, the newly formed viruses burst outward, completely destroying the host cell and spreading out to infect others. It's a pretty brutal cycle. The sheer variety of viruses in nature is staggering, and they're highly specific to their hosts. Let's look at 3 distinct examples to put this into perspective. First, HIV. This is an RNA retrovirus that specifically targets and infects human white blood cells, which eventually leads to AIDS. Second, hepatitis B. This is a virus that disrupts normal cell division in the liver and is actually linked to causing liver cancer. And it's not just humans either. The tobacco mosaic virus is a plant virus that requires a physical wound or a bug bite to enter the plant, which ultimately stunts its growth. Before we wrap up the non-living section, we have to briefly acknowledge prions. Now, unlike bacteria or viruses, a prion isn't a cell. And get this, it doesn't even have DNA or RNA. It is strictly a protein. Prion design susceptibility is dictated by complex mechanisms, specifically how a protein tends to fold into a beta structure and how it interacts with surrounding molecules through local side chain interactions. When these proteins misfold, they can trigger a massive domino effect of disease. And this is entirely based on physical structure rather than genetic replication. Wild, right? Section 4, Gram Stain Analysis Model. Visualizing cell walls. Okay, let's talk about the Gram stain. This is a real laboratory technique that separates almost all bacteria into 2 massive categories based on their cell wall structure, which is absolutely crucial for doctors choosing the right antibiotic. First up, gram-positive bacteria. These have a very thick layer of peptidoglycan and contain teichoic acids. Because this layer is so thick, it traps the stain, making them appear a deep, vibrant purple under a microscope. On the flip side, gram-negative bacteria have a very thin layer of peptidoglycan. But importantly, they have an extra outer membrane containing lipopolysaccharides, which act as endotoxins when the cell dies. Because of that thinner inner wall, they don't hold the initial stain and end up looking pink. Section 5. Misconception and formative checks. Testing your knowledge. All right, first up, a major misconception check. Are all bacteria harmful to humans? Well, think back to the examples we discussed earlier. The answer, of course, is a resounding no. While the harmful, pathogenic bacteria grab all the headlines, the vast majority of eubacteria are incredibly helpful. I mean, we literally need them to digest our food, to recycle nutrients in the soil, and to produce everything from yogurt to life-saving antibiotics. Let's tackle another super common misconception. Are viruses considered living cells? Based on what we just reviewed about their structure and behavior, the answer is definitively no. Remember, viruses do not grow, they don't have a metabolism, and they completely lack the cellular machinery needed to reproduce on their own. They are essentially just biological code wrapped up in a protein shell, and they are entirely reliant on a living host. Now for a slightly more detailed formative check. How does bacterial reproduction differ fundamentally from viral multiplication? Let's contrast those 2 mechanisms real quick. Bacteria are living, independent cells. They reproduce asexually all on their own through that process of cell division we called binary fission. Viruses, on the other hand, are non-living parasites. They multiply through the destructive lytic cycle, which means they absolutely have to invade a host cell, hijack its internal machinery to assemble new viruses, and then burst that cell open to escape. Section 6. Concise lesson recap. Wrapping up. Okay, here are your rapid-fire key takeaways for today. 1. Prokaryotic cells, like bacteria, lack a membrane-bound nucleus and reproduce independently via binary fission. 2. Archaebacteria thrive in extreme, ancient environments, while eubacteria are the common ones we interact with every single day. 3. Viruses are non-living. They're just a capsid protecting DNA or RNA, and they require a host's lytic cycle to multiply. 4. The Gram stain is a visual tool to identify cell walls. Thick walls turn purple and are gram-positive, while thin walls with an outer membrane turn pink and are gram-negative. And finally, 5. Prions are disease-causing proteins, defined by their specific beta structure folding tendencies. I want to leave you with one final provocative thought to really spark your curiosity. We just summarized how a virus uses the lytic cycle to hijack a host cell from the inside out, right? Well, since the virus uses the host's own machinery to build itself, how might scientists design medicines to stop a virus without also harming the host cell it's hiding inside? It is a massive challenge in modern medicine, and thinking about it really highlights just how incredibly complex this microscopic world is. Thank you so much for joining me for this explainer, and keep exploring the science all around you.